title of his presentation, let's read that. A controlled single-cell isolation platform and miniaturized sample preparation for single-cell omics and multiomics analysis. So about François. François received his PhD in 2012. I started my PhD in 2012 in the field of molecular and cellular biology. He developed his skills in genetics during 5 years in an NGO dedicated to genetics therapy for rare disease. There, François conducted studies to evaluate new strategies for gene therapy, focusing on RNA surgery. In 2017, François joined Selenium at the very beginning of the journey, taking part in the development of cell sorting and isolation process. Since 2021, he is a CSO and leads a team developing disruptive approaches to analyze cells on single-cell omics level. Please, François.
Thank you very much. Very nice introduction, very nice biography resumed. And then people will have time to move from one room to another. Awesome. I'm super happy to be there to present some of the solutions we provide to the scientific community around the idea of sorting and isolating cells for real, and then studying those cells at any kind of omics level, RNA, DNA, or proteomic level. My aim is to go through different applications, solutions we provide. The first one is the cell one, so that's a tool to sort and isolate cells. Then I will go through transcriptomic analysis in a miniaturized platform. We call that the cell and chip. We have the same strategy to study proteomics at the single cell level in the proteochip. And then some of our collaborators did work already on studying the entire transcriptome and the entire proteome from one single cell. And the tool is named nanosplits. But first, so a word about Selenium. The mission of Selenium is to provide disruptive technology, products, and services to better understand and master cell biology. Basically, we provide the tool, you do the biology. We have no clue of what you will do with these tools. The idea is to provide you what is the best on the market. So you can do the best developments of drugs, especially to treat people. We distribute worldwide. We have our flagship product, which is the Selenium one. So that's a cell sorter and isolator. I will describe how it's working. But just to let you know, this is dedicated to isolate cells. So we can go from 0.5 micrometer in diameter to 80 micrometer in diameter. So basically any kind of cells, from bacteria to very big cells, like cardiomyocytes. But to allow people to also handle group of cells, we have the Sphero one. So that's a tool dedicated to sort and isolate spheroids, organoids, or even small bodies. So a word about the Selenium one. Two things you need to understand. The first, we make a drop on demand. So you will probably think about flow cytometer, but that's not a flow here. We have a glass capillary where there is a cell suspension, and using a piezo dispensing system, we will just activate this piezo. There will be an acoustic wave traveling through the liquid, and then ejection of one drop. So that's drop on demand, drop in the air, and each time we make a drop, it's always the same drop, and we can do this drop when we want, where we want. So that's the first things, control the drop. The other one is that the detection of the cell is not signal-based, like in a flow cytometer. It's based on images. So we have a microscope in this device, and we'll just take images of the glass capillary, and the software will decide then to isolate or not one cell. And so to describe that a bit in more clear words, we go in the Selenium one. We have an open deck here, where you can put empty multiple plates or any kind of substrate, because you can dispense from this glass capillary in anything you want, or on top of everything you want. 384, 1536, that's not an issue for us. That's a very easy target. You can cool the deck, so you can control the temperature of the solution where you will dispense your cells. Another difference with the flow cytometer is that you load your sample, you aspirate your samples. So there is no risk of clogging, because everything which got in can go out. And then so the glass capillary is moving in front of this very big camera here. And the software will start to analyze this glass capillary and detecting this ejection area. Everything here will be in the next drop. there is a safety area called sedimentation area. And so you will make one drop here. The software will make one drop because there is no cell detected. One picture, one analysis, and then we'll move this glass capillary on top of one well and dispense this cell in well A1. Go back in front of the camera. Luckily, there is another cells to isolate. So after analyzing this picture, go back in A2. Back in front of the camera, no cell detected. You just discard this drop. Next one, doublets, you discard this drop, but actually you don't really discard those cells because they fall in a collection tube, so you can re-aspirate those cells if you want to be sure that you can sort every cells you have in your samples. That's dedicated for very precious cells. How it's working is it's basic image analysis. You detect the morphology of the cells, and if it's fitting the criteria we did define as cells I want to isolate, you will just isolate that. When you have doublet like this, it will not fit the elongation criteria, for instance, so you will discard that, or cell debris, like this one, will be discarded as well. So you make sure to isolate only single cell and the cells you want. And just to show that it's real, can you activate the video, guys? Sorry. Thank you very much. Oh, no, last one. Next one, sorry. Before this one. Again, before. This one. Here we go. So we are inside the cell N1, that's the glass capillary, ejection area. Each time there is a cell detected here, the head is moving and dispense the cell in the plate. We are in a 96 well plate, that's for cloning application here. When there is a doublet like this, first there is a red circle, and because there is a doublet, the stuff is not moving, and you discard the doublets. If there is a big cell, like this one, you don't isolate that because that's too big. You did define cells which are in the medium size, so you will only select the cells which are in the medium size. So that's the way we isolate cells with the cell N1. And on top of that, again, guys, sorry. You can sort the cells based on fluorescent signals, so that's still based on pictures. That's sequentially, we will take picture to detect if there is a green signal, live cells, if there is an orange signals, dead cells, so you can sort live dead like this. So each time the software will detect that the cell is green, not orange, then we will take a bright field images, check it's fitting the criteria of cell isolation, and we'll isolate those cells. So that's the way the cell one is working when you want to sort with fluorescent signal. When we say that we isolate cells, and we can isolate that anywhere we want, we can put a glass slide and dispense this drop containing these cells on top of a glass slide. So that's an array of 100 drops containing one cell per drop. You have the drop here and the cell inside, and you can have a look. When you want to isolate 100 cells, you isolate 100 cells. That's basically what you want to do. So just a quick sum up. With the cell N1, you can isolate any kind of cells from very, very small to very big, and actually very heterogeneous samples. If you have small and big cells all together, you can still isolate your cells. We are able to manipulate very low volumes, so starting from 1 microliter of sample is absolutely not an issue for the cell N1. And we are able to manipulate very low number of cells as well, from 1 to 1,000. So you will say, why do you want to isolate one cell? One is very low, but so we have customers who do cloning. They wait for the cells to divide, then they collect those two cells, and they isolate those two cells to do single-cell omics on that, for instance. Definitely, our throughput is not million. That's definitely thousands. A word again. So we can sort and isolate cells, but we make drop. And so we take advantage to this technology to have the same tool to sort and isolate cells and use it as a liquid handler and actually a nanoliter dispenser. And in doing that, so focusing only on sorting and isolation of cells, you can do cell line development. That's a super gentle technology, so there is no shear stress at all, meaning that if you sort live cells, they will grow. We have outstanding growing capabilities of cell sorting by the cell one. You can sort any kind of crazy cells. But using the capability of the cell1 to make very small drop where you want, you can also develop very specific omics. And I will go through that, showing that we did develop some solution for genomics, transcriptomics, proteomics, and multiomics. And to do that, because we make very small drop, I did not mention 500 picoliter the drop, so we are under the nanoliter. We did develop specific labware because we didn't want to work in plates in nanoliters. Basically, we did develop specific labwares to study DNA and RNA, and specific labwares to study proteins, because that's two different sample prep, very different sample prep. What about the one dedicated for DNA and RNA? We call that a cell-and-chip. That's actually 4x96 well plates miniaturized on a glass format. So that's 384. Very small wells, super easy to understand. That's a plate, but just a small one. Because it's so small, you need some specific tools to handle that. So to collect back the samples, you need to spin down funnels, for instance. But we provide everything, so it's very easy to use. And we use this chip to develop single-cell transcriptomics solutions. So that's a 3' RNA sequencing kit. We provide all the reagents with the chip, so you can perform the entire library prep from the cell isolated by the cell-N1 to the library ready to sequence. We did think of a very easy to use tool. So basically, we will provide a chip containing everything you need to barcode your cells. So there is already in this chip the barcodes, the lysis buffer, and the reverse transcriptase, meaning that you will have to focus only on the sample prep, and when you are ready to isolate your cells, and you can see the sample in the cell1, so you are sure that the sample really looks like the stuff you want to isolate. You will just grab the chip in the -80, put it in the cell1, and start to isolate your cells. And then you will just have to perform the reverse transcription in the cell and chip, which will introduce the barcode during the reverse transcription, and the TSO. And then you pull everything in the tube and you go for regular library prep with a cDNA amplification and fragmentation, and again, amplification of your library before going to Illumina sequencing. Super easy, and again, everything is provided. When you put the 3' kit on the market, now you have to compare to competitors because there is a lot of players on the market. So we did do this exercise. We did aggregate all the parameters you use in QC for 3'. So that's obviously the number of genes per cell, but the mapping rates, the fraction exon to intron, is that unique reads or not, the ribosomal contamination, percentage of deviation of dropouts. So we did aggregate all those parameters together on something which we call the performance score, and then we compare to a public data set. So that's coming from a publication dedicated to compare some solution on the market, and we did produce exactly the same data set, same cells, same cell states, same number of cells, same sequencing depth. Everything is similar to what has been published. And doing that, we are just above all the competition, which is currently on the market, and to make sure that everybody understands how sensitive are those technologies, that the SmartSeq 2, so that's the current gold standard for labs in R&D, and that the SmartSeq 3, which outperform actually everybody on the market. Like almost everybody now, because we provide a kit to produce data, and that's complex data, and I'm a biologist, and I'm not a bioinformatician, and I want to have the answer and not a crazy amount of DNA pieces on a text file. We also have a very easy-to-use tool called the Cell and Pipe. So that's an online tool, cloud-based. You receive some login numbers when you buy a kit, and you just have to drop the sequencing data online, and you will get a full report with all the QC and all the analysis on your samples. Super easy. There is absolutely no threshold button, nothing to change. I will go very quickly through the development of the whole genome sequencing applications. That's actually some collaborator who did that, French word, in BCCRC. The strategy is called DLP+ for direct library preparation, but I will not go through this today. That's basically the same idea of the transcriptomics. You just miniaturize everything in the chip, but that's for whole genome sequencing. I did not mention that we have a booth. I have some colleague here, so I will be pleased to present that if you have some question around this whole genome sequencing. But I want to move to have time to present that on the single-cell proteomics, which is for us, for me, I hope for this community, the next frontier in biology. So we have all the tools to do transcriptomics and to study cells now, but we know that's the intermediate picture, the final picture. What the cell is doing is the protein level, you know. So single-cell proteomics will definitely open a new avenue in biology. And why is it possible today to speak about single-cell proteomics? Well, that's mainly because of the key player in the field. So we work with Thermo Fisher Scientific and Bruker. The mass spec are, since a couple of years, a bit more maybe, sensitive enough to detect the proteins coming from one single cell. The big difference between transcriptomics or genomics and proteomics, you can't amplify the signal here. So you have 1,000 protein in your cell, you can analyze only these 1,000 of proteins compared to 1,000 of RNA. You will amplify that to billion of pieces of DNA. So thanks to this technical improvement, we are now able to do single-cell proteomics. But the challenge here is that it's so sensitive that every contamination you will introduce in the sample, you will detect it. And it may erase even the real results of your experiments. That's why we did develop some tools to work on the sample prep. A word first on the strategy you can use to do single-cell proteomics. There is already a lot of different strategies. You can regroup that on two different ones. You can multiplex things. So a bit like in transcriptomics, you will put some barcodes. Here is that's a chemical link to proteins. But you will put some barcodes, meaning that you are able then to do the mass spec on several cells together and recover who is coming from which cells, basically. So that's a multiplex one, fitting mainly the Orbitrap technology. And you can do the label-free technology. So basically, you will really do the sample prep cell by cell and go to the mass spec cell by cell. And this technology is more fitting the timsTOF approach. You can see that there is a lot of steps, especially for the multiplex one. So what we provide using the CELN1 and this specific CELN chip is a way to convert your cell to the sample you will have to inject in your mass spec in a fully automated manner, meaning that you reduce the risk of contamination. So the workflow you can run in this chip, which is dedicated for the multiplex format. We call that the Proteo chip 1216. That's 12 times 16 wells, because you can multiplex up to 16 cells currently in proteomics. That's quite low when you think about numbers in transcriptomics and genomics, but that's currently the status. A bit more, to be honest. That's 8 I think now. So we will probably change a bit the format of the chip. But so everything is included in the kit we provide in the solution by the CELN1, meaning that you will be able to do the lysis, the cell isolation, of course, TMT labeling, which is a very tricky part of the protocol, the quenching, and then you will pull everything and you will go directly to the autosampler because it's compatible with the autosampler. If we go through one publication using this strategy, and actually that's the guy who did develop this with us, Altmaier and all, they did some technical demonstration. So they did work on HeLa cells and HEK cells. You have here the PSM, peptides, and proteins, meaning a bit the reads, mapped reads, and number of genes per cell, for those who are, like me, more familiar with RNA than protein. What is important here is that you detect 1,500 proteins for HEK and around 2,000 proteins for HeLa cells. That's a lot. That's probably a good representation of the proteins expressed by those cells. Running this kind of experiments, most of the time you have to put with your single cells kind of a mock carrier, let's call it like this. And so you lose some space when you run the mass spec. In this demonstration, they just show that the carrier is useless. You can run directly the single cells. It's working well. Here you have some that's a triplicate of experiments, so just showing the overlap between the triplicates. And then you switch to more familiar representation for transcriptomics people. That's a kind of a t-SNE representation, and you will have the cluster of HeLa cells and the cluster of HEK cells, which are actually two different stages of cells. And you can have a volcano plot as well, meaning you have cells differentially, proteins, sorry, differentially expressed in HEK cells or HeLa cells. So that's basically all the information you can gather from proteins. All the strategy you have for transcriptomics, you can apply that for proteomics data analysis. So key message here for this multiplex approach, up to 2,000 proteins detected per cell. A very, very, very good signal-to-noise ratio, meaning that you don't have to use carrier, and that's actually the very first demonstration that you don't need any carrier to run multiplex samples in single-cell proteomics. If for some reason you don't want to run multiplex, or the samples don't fit the workflow, or you don't have the proper mass spec to do that, we also provide a solution for label-free. So one by one, this chip is just dedicated for this label-free protocol, meaning that you will do the sample prep cell by cell. The workflow is a bit shorter, and then you will load the sample cell by cell in the mass spec, which is in this case a timsTOF for single-cell proteomics. That's how the chip look like. So that's, again, very easy to run this protocol, and you reduce the risk to introduce some contamination by far than doing that by hand in your labs. And again, some very basic technical verification. What kind of results can we get with this workflow? So we did isolate 1, 5, 10, or 20 cells to see the sensitivity, and there was kind of a plateau effect on HEK and HeLa cells, or the same cells. And in this case, we have the result for 1, 5, 10, or 20 cells, and you can detect 800 cells on HEK and 1,100 for HeLa cells. So a bit less proteins than in the multiplex workflow, but there is good reason for that. I mean, the database of reference, the ways you are analyzing the data, you need to consider that we are at the very beginning of proteomics, so that's basically the very beginning of transcriptomics. And so the reference database play a crucial role in the number of proteins we will detect through the dataset. So basically the take-home message in this label-free workflow is 1,000 of proteins per single cell. That's designed to increase the number of peptides in this case. We are super happy to play with most of the key opinion leaders in this field. So Karl Metzler, we are working kind of a daily basis with him. That's the guy and the team who did develop the Proteo chip for the multiplex workflow. We are working a lot with PNNL and Yingzhu. We are also working a lot with Nikolai Slavov, who did develop another approach. And we work also with Cedar Sinai and Broad Institute. So we are very happy to be part of this very important game. Real quick, before questions, you can do transcriptomics, you can do proteomics. Why not doing both together? Real transcriptomics and real proteomics. No antibodies and no targeted qPCR or something like that. Using the CELN1 again, what you can do is to dispense your cells on the glass slide, and then you can use again the CELN1 to dispense the lysis buffer or whatever you need on top of those cells, meaning that you will have this cell lies in this case on top of a glass slide, and then you can come with another glass slide in front of this small droplet and just touch like this, and so you will basically mechanically split the drop into half a drop, meaning that then you can take this one and go for RNA sequencing, and take this one and go for single-cell proteomics. So you will have half of the content of a cell in each drop, and you expect to have good results for transcriptomics and proteomics. Do we? I mean, do they? Because the job has been done by PNNL again. Again, 1 cell, 3 cells, 11 cells, looking at RNA-seq level, proteomic level. If we focus on what we want to focus on, that's a single cell, you have up to 6,000 genes detected and 3,000 proteins detected per cell. So that's a pretty encouraging result, I think. So Maybe here, that's the beginning of the answer of what is happening between RNA and proteins, you know? Because there is a lot of regulation between pre-messenger RNA actually, and real proteomics. And if you have both data sets from one single cell, then you start to have the tool to really understand the biology behind, I think. So if you want to have more information about those technologies, please come visit us at the booth. Of course, I thank all the people who did work to develop those things. I thank all the customers who already trust us, and that's a lot already. And I take any questions.
Thank you, François. Any question, guys?
Can I maybe ask which sample type you use to perform the single-cell transcriptomics experiments?
So single-cell transcriptomics,
Yes.
3' RNA sequencing.
Yes, you showed some data, but you did not really provide which type of sample.
So that was HEK cells because we wanted to compare on public data sets. But we use this kit on any kind of sample, skin, cancer, yeah, cells from the blood, anything.
Thank you.
The time cost and the manual time to process what?
Let's say 1,000 cells, yeah. How long will it take on your machine and how much money?
How much money? So for transcriptomics, we have the product manager here.
Sorry, for MS?
For MS, so the kit is pretty cheap. Then the mass spec is pretty expensive. But you need to complain to Brooker or Temo then. I'm sorry for the price. I'm just a scientist. Sorry. So.
Any more questions? No? Thank you, François.
Thank you very much.
Thank you.